Well, welcome everybody. I am Carnegie Science President. My name is Eric Isaacs, and I want to welcome those here, our friends, supporters, and interested parties here to the Martin Luther King Jr. Memorial Library, but I also want to welcome those who are online and joining us virtually. I just want to say a few things about Carnegie first. You know, Andrew Carnegie was very early in the business of ecology and ecological systems, and in fact, one of the first two experiments that he funded in 1902 was to study the saguaro cacti in the Sonoran Desert in Arizona. And why did he want to study them? Well, why did someone want to study them? Because they're plants that exist in extreme environments and come back to life when it rains and kind of sit there living without any water for decades. It's an interesting problem, but it's something that we've been doing for a long time. To this day, we do ecological research. And the eye we take toward this is going to be a lot like what you'll hear about tonight, which is to look at the molecular basis, the genetic basis, for how things live, how they interact with each other, and how they provide massive amounts of support for all life on Earth. And you're going to hear about that tonight. So I'm really excited to hear what we have. Tonight we have the Fondation Tara Océan. Did I say that right? The Tara Ocean Foundation, and it's a very interesting organization that, as you don't see it in the picture yet, but that has been studying the oceans for quite a while. Why do we care about the oceans? The oceans are extremely important because, first of all, our web of food is intimately and integrally connected to everything in the ocean, particularly to the small, the microbes, the plankton, the phytoplankton. Those things are really important for the web of food, but also the air you're breathing. About 50% of it comes from the ocean. So, and I'd say about 40% of the ocean of the world's carbon also gets fixed and dropped to the bottom of the ocean out of our way. So it's a very important ecosystem, and it's vast. It's the biggest life ecosystem in the world, and it's very little understood. It is just now becoming better understood, in part because of what Tara Oceans is doing. The one thing I'm going to introduce our speakers in just a minute, but in particular, Tara has done exactly this DNA studies, the molecular studies of the vast amount of different kinds from phytoplankton and plankton to, I think, the diatoms, everything that's in there. You'll hear more about that tonight, and I won't go into detail, but this kind of study is extremely important, as I've just told you, because of the level of what it does for us humans, but also what it does for all life on Earth, including plants on dry land as well as in the ocean. So what I'll do now is I'm going to introduce we have three affiliates with Tara Oceans, two who are in Tara Oceans and one who is a user of their data. They produce vast amounts of data from all over the globe. All the oceans are around the Earth. And first, we're going to hear from Romain Troublé. He's the executive director of Tara Ocean Foundation and president of the Ocean and Climate Platform. You may recognize his name because he was also a competitor in the America's Cup in 2000 to 2003. Is that correct? So that's another interesting history in his love of the oceans, obviously. He's worked in polar logistics and for both supporting scientific missions and discovery of a frozen mammoth in Siberia. So obviously a lot of experience in this kind of science, and we're looking forward to hearing from him. Next, you'll get a chance to learn something from the scientific coordinators of the Tara Oceans Project, Dr. Chris Bowler. And Bowler is the director of the research at CNRS. Many of you know CNRS is kind of like the Bell Labs of the French telecommunication system. I collaborated there often. And also director of the Ecology and Evolutionary Biology section of the Institut de Biologie de l'École Normale Supérieure. And that's in Paris. So it's a great place to live. If it's not New York, it's okay, it's Paris. And then last, we're gonna hear from Dr. Matthew Sullivan, who's a professor of microbiology and the director of the Center of Microbiome Science at the Ohio State University. And he studies coevolution of microbes and viruses, two very integrally related. Of course, microbes exist in all of us. They exist all over everything, and there's nothing that lives on Earth without the microbes and the viruses. They're good and they're bad. They're in your gut, they're on your skin, they're everywhere in the ocean. And he studies these through micro-mediated biotechnology, bioenergy, et cetera. So, and if you look at the list of publications on his CV, you'll see more than a dozen that include Tara Ocean. So he's a great user of the data. So we have here the director, we have someone who's intimately involved in the programs, and then we also have someone who's using the data to hear from. And I'm really excited to hear what they have to say. Just a brief sort of more clerical note. You'll hear it from the three of them. And then we'll have Dr. Troublé. Troublé is going to come back up and show us a video. and then we'll take questions. We're using Pigeonhole. You see the sign here. You can, maybe it's not there anymore, but you'll use Pigeonhole, and you'll be able to put your questions, and I'll be able to read them, and I'll be able to ask these three scientists the questions through that direction. So if you'll allow me, let's welcome our three speakers tonight.
Hello, everyone. Good evening. Thank you very much, Carnegie, for having us tonight. It's a fantastic opportunity. And thank you for coming. Thank you for coming also for those who are online today. So my name is indeed Romain Troublé. Trouble. You are in deep trouble tonight. Be careful. Yeah. So we work on the planet, and we work at the global scale with this very small schooner, Tara, 36 meters only, with sails, sailing around the world to understand better this planet. And from space, this planet was really blue. It's a blue planet. And when you really look at the quantity of water on this planet, it's not that much relatively to the size of our planet. So we really feel that the ocean is endless, very deep, and so much water. We can put everything we want into it, and we can take anything from it without having an impact at our scale. But in fact, it's not that much. And life appeared in this small ball of water that you see here. And life is sustained, as Eric just said it, very much by this ocean and the healthy ocean that is we have today still. So 20 years ago, we decided to go and to launch this foundation. Agnès B and her son Etienne Bourgois, the founders, they decided they're not scientists at all, they are fashion designers, but decided to put some energy into the ocean. Because of the reason that Eric also said just before, 71% of the planet is covered by water. It's huge. I'm a sailor. I like to sail on the water. It's so big, so nice to go everywhere. Also for the oxygen production, the carbon dioxide, the heat that the ocean is taking from the atmosphere to the bottom of the ocean is really our life support system today. And what is our life support system is the life that is sitting into it. And it's really what we care about the TARA Foundation since 20 years. So in France, it's very famous. The TARA Foundation is the most famous. We have NGO, non-profit in France. We own this boat, the TARA vessel that you see here. It's not very big, it's very small, but it's very efficient. It's a little boat. It's the little boat that could, this little, yeah, kind of the same way. And there's two missions: is to explore the planet as much as we can, and to share this scientific knowledge to trigger change, to convince people to move, to convince them as well that there is climate change on this planet. And the ultimate goal is to protect the ocean, to save this ocean, of course. And the core of what we do is biology. It's life. And those two stresses that is affecting life today on the planet is climate change and pollutions, mostly. Of course, our activities as well, sometimes. And this project is about exploration. It's nice to explore the planet, to show people that we need to explore much more. Space is important, but ocean is also very important. Outreach and also policy. We had a lot of energy putting at the UN, United Nations, and partnerships. And if I'm here today with Chris Boller and Matt Sullivan, it's because we are in partnership with public scientists across the world. We don't have, I'm a scientist myself by training, but we don't do science in the foundation. We work with laboratories across the globe. Before, I don't want to talk too much. I want to take you on a tour, so I will show you some footages, just a 3 minutes video to take you far away from here. A good way to take off from the Washington. Yeah. And so it's a partnership, I said, and partnership with also a lot of partners that are individual, but also companies that are supporting us since 20 years. And I'm doing this work since 20 years with the founders. So it's kind of a big chunk of my life so far to put into this project. So we have done 13 expeditions so far in 20 years. 20 years, we just had the birthday 3 weeks ago. So it's really the 20 years. And we crisscross the planet, as you can see here, sailing about 600,000 kilometers and visiting 75 countries, doing research in these countries. Again, partnerships. We must do that. And tonight, we're going to speak to you about a particular mission that took us for 4 years around the world to do this track, to sample the planet by every dot you can see here, to sample the ecosystems, the viruses, bacteria, phytoplankton, protists, zooplankton, the bees and the insect of the ocean, but also the viruses that Matt knows very well. And so I will not speak too long and give the floor to Chris, but this project has been launched by a researcher, a cell biologist, very far from an oceanographer, a cell biologist called Eric Carsanti, and I would like to just pay a tribute to him tonight because his vision was really a very crazy vision back 15 years ago. Sorry, and just to show you this impact, and we're going to talk about it again, but this result will be explained by Matt and Chris, but to show you the impact of such a project with such a small boat, with a few folks, because we're not much of us, how much we have done with the science we have produced. And we have published together 200 scientific papers, and the data that went public, open source, triggered 900 papers over the last 7 years. So it's a lot of impact. On this, I would like to come on stage Chris Border, my dear friend and long-term partner and crew member. Thank you.
Thank you. Thank you, Romain, for setting the stage so nicely. And thank you to all of you for coming here, braving the cold evening in Washington, D.C. So I'm going to tell you a bit more about what we've discovered about life in the ocean through these different projects that we've had with Tara Oceans. If we think about life in the ocean, you may think, well, where is the life in the ocean? You know, if you go out for a walk in the forest, it's easy to see the life all around you, right? The animals, the plants, the trees, and so on. But in the ocean, where is the life? You may think the ocean is barren, but in fact, not at all. You just need a microscope to find this amazing rainbow of organisms that are present in this invisible world that we call the plankton in the ocean, that do all kinds of important things for us. So these are very, very small organisms. They're single-celled by the most part. We can consider them the microbiome of the ocean, looking after the health of our ocean. You can get lots of these microbes on a tiny pinhead, as you can see in this image. And although they're very small, they are incredibly diverse. Here we have the whole tree of life of eukaryotes on our planet. These are complex organisms that have cells like ourselves. So we don't have bacteria or viruses on this slide. We have the eukaryotes. On this slide, we have plants, which are shown sort of up here at around 10 o'clock in this branch of the tree. Down here, we have ourselves, the animals. We're sitting within the animal branches down here. And our closest neighbor in this tree of life are the sponges, okay? So the distance between us and the sponges are this tiny branch at the tip of the tree here. In addition to that diversity, you know, in addition to the diversity between animals and sponges, is all of this incredible diversity that you see in this tree of life, going back, to the very origins of life on our planet. And all of this diversity is essentially present in the ocean. So in a single drop of water from the ocean, you basically have the tree of life sitting there waiting to be discovered. And this is one of the things that we've been working on over several years. And Matt will allude to that a little bit later on during his presentation. So we have beautiful diversity of life, and we also have microscopic organisms that do incredibly important functions for the well-being of the ocean. These organisms provide the food that feeds all of the larger organisms, the basis of the ocean food webs. These organisms, many of them that we call phytoplankton, are photosynthetic, and they are sort of the equivalent of the plants on land doing photosynthesis, but they're not, you can't see them, they're microscopic. And through photosynthesis, they take carbon dioxide out of the atmosphere, they convert it into life, into themselves, which then feeds the food webs to the higher organisms up to ourselves ultimately. Through photosynthesis, of course, they also generate energy. They also generate oxygen, which is also kind of important for a lot of organisms, and for the current state of our planet, oxygen is extremely important for the survival of many, many organisms. Now, an additional point with the ocean is that because the ocean is deep, when these organisms die, a fraction of them will sink to the bottom of the ocean, taking the carbon with them, and ultimately generating fossil fuels. The oil and the gas that we so happily burn today is plankton, okay, that has come down from the surface of the ocean, sunk down over millions of years of geological time, and generated these oil and gas reserves that we are now consuming. So this is one way in which the plankton interact with the climate, by taking carbon dioxide out of the atmosphere. And because these are living organisms, they're also very sensitive to changes in climate. And so there's a sort of two-way feedback between the climate and the plankton that we need to understand more in order to understand, how the ocean is going to change in the future. So it's a complex world. You can get an idea of the complexity of these organisms through that slide that I showed you. It's a complex world, it's a microscopic world, invisible world to the naked eye. So how do we try and disentangle it through this project that we've been talking about today? Basically, what we want to do is to try to understand who is there, what are they doing, with whom are they doing it, and why are they doing, all of the different kinds of things that they're doing, okay? Why is it important for them? Why is it important for the ecosystem? Why is it important for the functioning of the whole Earth system? So we basically want to try and figure out this rather simple question, but, trying to disentangle it in an invisible world that we cannot see. So in order to do that, we need to sample the entire community, from the very smallest players, the viruses, through to the bacteria, the prokaryotes, through to the smaller animals. So to sample all of the community so that we can see the whole thing together. We need to sample this globally because the ocean is connected, right? Everything can be everywhere else, a week, a month, a year later. So we need a global coverage for this sort of project. And we need to sample in a very standardized way. And we need to analyze our samples in a very standardized way so that with this global data set, we can compare one point with everywhere else. So, we can compare what we find in the Arctic with what we find in the Antarctic, with what we find in the Maldives, with what we find in Chesapeake Bay, and so on, all right? So by standardizing our sampling all of these different organisms, going from the surface down to 1,000 meters of depth in the water column, sampling day and night, we try to ensure this uniform sampling. And then once we have the samples back in the lab, we again use standardized analytical pipelines to analyze them based on high content, high throughput microscopy, and based on a lot of DNA sequencing to try and figure out, disentangle this complex community. And so essentially using a sort of systems approach to try to understand this complex system at a planetary scale. This was the general approach that we used in this project. And we began publishing our work, our first findings from the project already now back in 2015, where Science gave us a special issue where we published 5 papers describing the first reports, the first results from this large sampling program that involved like 30,000 samples that were analyzed at the end of the project. So we have this beautiful data set describing, you know, the ocean. Now, what can we do with it? I'm sort of going to take you through some a few different ways in which we've used this data set to learn more about the ocean. Really beginning with some very basic questions, and through this you will see that, yeah, how little we really knew about the ocean before you know, undertaking this big global program in the first place. So very basic questions that an alien coming from space might ask us, you know, with whom do we share our planet? You know, in the ocean, which organisms are the most abundant? Which organisms are the most diverse? We should know that, the answer to those questions, right? And you'll see the answers are quite surprising. But before I show you the answers, let's look at the answers to those questions in an ecosystem that we can better understand. Our terrestrial ecosystem, you know, when we go out and we look around us, what do we see? You know, not necessarily in Washington, D.C., but when we get out of the cities, we see that the land is green in principle, and for the most part, right? That is because it's the plants, the photosynthetic organisms that dominate the biomass of life in terrestrial systems. The plants really dominate the abundance of organisms. And we're down in the bottom corner within animals. You can see that we're not particularly significant on with respect to the plants. So in a terrestrial ecosystem, it's the plants that dominate in terms of abundance. Now, in terms of diversity, the answer is really quite different. The answer is shown on the right side of this slide, and the organisms are shown. Their diversity is represented in terms of the size of those different organisms. And you can see here that it's the insects which are dominating in terms of diversity. These are the most diverse organisms in our terrestrial systems. So the question I'm asking really is, who are the plants and who are the insects of the marine world? And the answers in both cases were a surprise even to us, actually. In terms of the abundant organisms, the most abundant are organisms called radiolarians. I'm not sure if you've heard of radiolarians. We've known about radiolarians for a long time. You can see these beautiful images of Ernst Haeckel that he generated more than 100 years ago, which actually stimulated the Art Nouveau movement and sort of organic architecture in general. These are very beautiful organisms, single-celled organisms that you can see there. We never really appreciated their abundance because they're very fragile, they're difficult to sample, and so on. But through our program, we identified the radiolarians as the most abundant organisms in the ocean. In terms of diversity, the most abundant organisms are called diplonemids. How many people have heard of diplonemids? Nobody, right? Neither had we. To be quite honest with you, diplonemids, we still don't know why these organisms are so diverse. We don't know much about them because we don't have organisms that we can use experimentally in the lab. But because they're sitting in the tree of life, sort of down in this bottom right-hand corner, together with trypanosomes, leishmanias, these sorts of human parasites. We assume that these are parasitic organisms parasitizing on the other plankton groups, but we don't really know what their lifestyle is. So these are the insects of the ocean, okay, the diplonemids. So you can see that even with these very, very simple questions, we get very surprising answers. Now let's dig in a little bit deeper into the data set. So as I mentioned, we were interested to sample the whole breadth of organisms within this planktonic world, which in a world that we can understand would translate to sort of sampling everything between an ant and a brontosaurus in a forest, okay? So we have all of these organisms going from the very smallest viruses up to the largest sort of small animals in our data set. We have all of this data. We have it from multiple sampling sites around the world, and we have it from different depths in the ocean. So we can take all of this data, we can throw it all together and do a giant compute and ask, well, which organisms co-occur with each other? Which organisms do we always find together? Or which organisms do we never find together? So through this computational exercise, we can sort of define the social network of the plankton, the Facebook of this plankton world, if you like. And this is a sort of hairball which emerges from that exercise. It's not very pretty to look at, but it's incredibly interesting to dig into, as you'll see in a second. So each dot on this hairball here is a different organism, okay? They're color-coded by different kinds of organisms, whether they're bacteria or eukaryotes and so on. And then the lines between two organisms indicates whether they co-occur. So the green lines indicate that these organisms are found together, or the red lines between them indicate that they are never found together in our multiple samples, okay? So we can begin here to define, you know, some organisms that are sort of very social, you know, they have lots of green lines around them. These are sort of the very popular, the social species of plankton, the sort of the Beyoncés, the Taylor Swifts of the plankton world, okay? And then we have these antisocial guys with a lot of red. They're more on the right side of this plot with a lot of red lines around them. These are the unpopular forms of plankton that people don't want to hang around with. I'm not going to make analogies with who they may be, especially not in DC. But but you can see there's a lot of, you know, detail in this sort of plot. And if we disentangle it just simply by pulling out, you know, the number of positive associations with the number of negative associations, we can see that the number of positive associations really dominates, implying that sort of in this world of plankton, it's cooperation that is more important than competition. This was another surprise to us because, you know, through our neo-Darwinian education, we think a lot about survival of the fittest, competition, the guy who is strongest wins and so on. But, you know, the lesson perhaps from this plankton world is that, well, perhaps life is more about cooperation, more about working together to ensure that the whole ecosystem works together and works, you know, to the benefit of everybody. And this perhaps is a more useful ecological strategy than the sort of screw your neighbor kind of strategy that that is perhaps more what we would think about from a neo-Darwinian perspective. So that was an interesting lesson that we also got from this work. So that's the sort of big pattern, you know, that we see, a macro scale pattern that we see from looking at this this interactome. What we also can do here, we can dig down and we can find our favorite organism and see, well, what does our organism look like? How does it behave in this network? So this is a sort of playground to discover new interactions between organisms. And I'll give you one example of what we could figure out from that sort of exercise by working with this, studying this organism called Symbiodinium. It's a dinoflagellate. We know that this is a sort of a super symbiont because we find it in symbiosis with coral reefs. We find it in symbiosis with mollusks and other sort of sea animals. It's responsible for a lot of the color in the ocean because it's a photosynthetic organism. So we know a lot about the symbioses of Symbiodinium. The question we can ask here is whether we find Symbiodinium also in symbiosis in the planktonic world, you know, floating around in the ocean. And we're very surprised to find that, yes, it's also in symbiosis when it's floating around in the ocean. And it's found a home. It's found a home inside another organism. So another symbiosis going on inside this organism, which we call a ciliate. It's a single-celled organism, maybe 1/10 of a millimeter in size. It's got hairs all around it, as you can see from the green image here. And if we look inside this ciliate organism, The little red balls, the red and blue balls, are the symbiodinium cells living inside this, this home that it has found. And we're very surprised to find this, sort of globally in our data set. And, so presumably symbiodinium, it doesn't just float around naked in the planktonic world, but it lives inside this home. And when it finds a suitable coral reef, perhaps it jumps out and then starts to colonize the corals as well. So that's another surprise that we get from this sort of work. So these are some of the ideas that have emerged from the different studies that have been performed over the years by the Tara Oceans Consortium in particular. And those are just giving you a taste of the sorts of things that we have found. We're very satisfied to get recognition of our work recently from Nature magazine, who published a little editorial about us in the 8th of June issue of this year, talking about Tara's inspiring 20 years at sea. So that's a great recognition for scientists to get that sort of recognition of our collective work over these years. So I cannot really conclude this part of the talk without going into a bit of detail of what we can learn about climate change from these studies, because this is obviously a burning issue for many of us these days. So, as we all know, we have become incredibly powerful as a biogeochemical power on our planet. We are making a lot of changes to our ecosystems, and we can really see this also in the ocean. When we look at actual data coming from the ocean, in the top left, you can see how the temperature of the ocean has increased since 1900. The top right, we see the sea level rise since 1900. The Arctic sea ice in the summer has lost about 50% of its surface in the last 50 years. And the oceans are slowly acidifying because CO2 plus water makes carbonate, bicarbonate, which is acidic. So all of this is actual data where we can really see, through data, not simulations, not models, but actual data, we can see how the ocean is changing, really in front of our eyes. So, with this global baseline of data that we have from Tara Oceans, what can we learn about the impacts of climate change on the ocean? I'm going to give you two examples of this. One where we have sort of dug into the past to see evidence for the effects of ocean acidification over the recent history of the ocean. So to look for the effects of ocean acidification, ideally, what we would like to have is samples from the past, right? Ocean samples from the past before the ocean was too heavily impacted by humans, so that we can compare the ocean today with the ocean how it was, a couple of hundred years ago. This is pretty difficult because there are very few samples like that around, but we are lucky to have a set of samples from the HMS Challenger expedition from the 1870s. This is considered to be the first oceanographic expedition, where for 5 years they went around the world looking at ocean life, maybe mainly life in the deep ocean, collected samples, and those samples, many of them are now stored in the Natural History Museum in London. And so what this study was interested to look at was whether acidification has impacted the process of calcification of a group of organisms called foraminifera. Now, we know that the calcification process is sensitive to pH changes, so we're interested to see whether these foraminifera have changed in the period of time between sampling on the Challenger and sampling on Tara. In particular, the forams have these calcified cell walls, which we can see labeled in blue on the right side of the figure, and in the bottom right, you see the thickness of those cell walls in samples collected by the HMS Challenger, and above you see samples collected by Tara, and you can see really the differences are really quite striking. Today's foraminiferas have much thinner shells compared to those in the past, and this is one of the first real direct evidences that we have of the actual impact of acidification on the ocean, on life in the ocean. And finally, in addition to digging into the past, we can try to look into the future to see what's in store for plankton as the oceans change in the future. So this is possible because thanks to the Tara Ocean sampling, we have a global sampling, consistent uniform sampling, where we can generate global maps of the distributions and diversity of different kinds of organisms within this planktonic world at a global scale. And you can see that for 6 major groups of plankton in this plot. So this is sort of the state of diversity of these organisms today in the ocean, beginning of the 21st century. With this data, we can inject this data into the models of the IPCC climate panel, who have made simulations about how they project that the ocean will change by end of 21st century in terms of increases in temperature, changes in photosynthesis, productivity, and so on. So we can inject today's plankton into this future ocean and see what happens. And here we sort of see the result of that exercise, where we're looking really at the anomalies of diversity of these different groups of organisms by end 21st century. Basically, any color that you see here indicates an anomaly, a change in diversity of this group of organisms. And what is perhaps most important to see is when we plot those anomalies by latitude, going from Arctic on the top through equator to Antarctic at the bottom, then we can see that those regions with the highest changes of anomalies are areas where we have high levels of carbon export, so areas where carbon dioxide is taken out of the atmosphere into the ocean at high level. These are areas where fisheries, our fishing industry is particularly active, and these are areas of high levels of marine protected areas where we actually try to protect the diversity in the ocean. So these are potentially areas that are going to be heavily impacted by these changes, these anomalies in biodiversity as we move into the future. So I think I will end there with a summary of what we have done with this baseline of data from today's ocean, and how we can use it in many different ways. And I will hand over to Matt to tell us a lot more about the viruses.
Great. It's so exciting to be here. My story for Tara is actually, I was a substitute speaker at a conference in Monaco, and Chris gave a talk about 8, I think, scientists at the time who had a sailboat and were going to sail around the world. And I just started a research lab as a faculty member, and I thought that sounds really cool. That would be great. And so Chris let me play. So we're going to talk about some of the virus stories, and I'm really a virus of bacteria expert, so phages, and this is the world that they see. That's a T4 phage, the lunar lander type virus that infects E. coli in your gut. And at the very end, I'll give a teaser about RNA viruses and giant viruses that might come closer to home to what you normally think about for viruses. What our picture was when we started was this. If you concentrated seawater, you'd see big dots and little dots. And we could count those. The small dots are viruses and the bigger dots are cells. And you could see that the small dots outnumbered the cells. So viruses outnumbered the cells, but I was very unhappy with that because I wanted to know which of those dots infected the larger dots and what are their stories. And so we developed genomic tools to systematically and quantitatively study these. And then we started creating these vast catalogs of viruses on the planet. So we created a global catalog throughout these oceans. And it was complicated because 99.9% of what we saw was totally new to science. I couldn't put a name on it. I had to figure out a way to categorize it. So we developed, and at this time, this was pretty novel, machine learning approaches to organize that sequence space to be able to even give those viruses a name or at least a category. When we try to think about the state of science then versus now, this was all of the viruses that infect microbes that were genome sequenced. So each of those virus pictures there represents 1,000 virus genomes. We expanded it rapidly with the first round of expeditions and even more with the second round of expeditions. And we've got numbers now that are phenomenal. Instead of 2,000 virus genomes, we now have hundreds of thousands of virus genomes from around the planet. And in fact, I use the word species here because we formally looked at that and determined that these genomes fit a species definition, a biological species definition. So who is there? It turns out the viruses that are most abundant in the oceans are T4 phage. This is the most abundant virus genus. T4 might be something you've heard of. It's the virus which is the basis of molecular biology and genetics. A lot of its enzymes are used in molecular biology. You've probably not heard of 538.1, but this is the third most abundant virus genus in the oceans. And it's now probably the most well-studied virus on the planet. We've done time-resolved multi-omics, heavy modeling, all kinds of stuff to study how this virus infects its hosts under different conditions. So just getting catalogs helps us learn who's out there. It also helps us to start to track these viruses. So this is focusing in on a particular region of the Tara Ocean's global track. Gray dots are where the boat stopped for 24 to 48 hours. And the red lines or blue lines or green lines, these are all the flow or the genome tracking. Where did the viruses go from site to site? And what we learned is that the viruses just ride the currents. So that's a pretty expensive experiment to tell us that viruses float. It turns out viruses have a low Reynolds number, and we also saw that they sink. And so did you see the X's come on here? A lot of those sites showed strong evidence of viruses sinking. That was the big surprise. And it was a big surprise because of this. The paradigm was cells get infected by viruses, they blow up, those small cell parts get rapidly recycled because the oceans are very limited for nutrients. We're going to vastly recycle. That's called the viral shunt. But we're seeing viruses sinking. They should not sink if this is happening. What if instead those local virus explosions led to aggregates, and those aggregates could sink out of the water column with viruses stuck in them? Is that what we're seeing? Or could it be that viruses actually change the biochemistry, the pheromones that cells put out, so that they're actually attractive to grazers? Then the grazers, fecal matter would sink the viruses out. So this was an alternative view of how viruses could impact carbon cycling in the oceans. And the exciting thing is in Tara, every paper has many co-first authors. So this is 4 co-first authors, which were applied mathematicians, oceanographers, ecologists, and they were trying to understand if they could use sequence space, computational approaches, and rules about how ecosystems work to develop a predictive modeling genes to ecosystems context. And the question they were interested in was this: which organisms drive carbon export in the oceans? And Chris just mentioned how important this carbon export role is for the oceans. The oceans buffer against climate change by about half. So they're protecting us against the damaging elements of climate change. In this plot, which was Tara's, for the first time getting a global map of carbon flux with this resolution, the blue areas are low carbon flux, and the red and yellow are areas of high carbon flux. So we just were asking the question across all these many organisms that we study, 8 orders of magnitude of ocean biota, which organisms best predict carbon flux? Now, I'm the virus guy, so the story was viruses were the best predictor. In fact, they predicted 89% of the variation in carbon flux. And in ecology, if you predict 89% of variation of anything, that is really, really strong because ecology it's very complicated. So this suggested that this paradigm wasn't right, and the alternative hypothesis must be happening. Viral lysis must form these aggregates, leads to sinking out of the water column. And so we've actually, a whole new field has started just looking at using experimental approaches now to test these kinds of virus-mediated exports out of the oceans. So we can count these dots, and we can do this quantitatively. We can assess their genomes. We've now done a study to look at carbon flux. The other thing I didn't tell you about being able to answer this question is we can come up with these VIPs. So not only are we figuring out that viruses are important, but we can figure out which viruses are important. And that allows us to, if we're being a PhD student and we want to study one organism for 5 years, which organism should you study? And so now with Tara, we can say, study the VIPs. And so we've developed what we call virus cell experiments, where we look at time-resolved infections, multi-omics experiments, and do studies that'll look at infection efficiency, how a virus, when it's infecting a cell, is impacted by being grazed or not, how two different viruses infect the same cell, and how that changes when you modulate nutrients or how the cell can grow. And so these virus cell experiments are actually the basis of what we hope will now drive global ocean predictive models by now including the virus-infected state. Another mystery that came up was this thing called AMGs. When we looked at virus genomes, we saw that they had AMGs. AMGs are auxiliary metabolic genes. What this means is viruses have stolen a gene from the host cell, and they've used it to their advantage. And this was actually well known before Tara. This is a group of many papers that had studied cyanobacterial viruses. These are viruses which infect small photosynthetic cells. And the viruses have photosynthesis genes. This has been such an important element of viral biology that Paul Fokowski recognized it as one that could impact planetary chemistry because these viruses were infecting globally distributed cyanobacteria. So what is happening here? This is the photosynthetic membrane where you have two photosystem reaction centers. A photon of light strikes the first photosystem, which happens to be called PS2. And then it can strike, a photon of light can strike the second reaction center, PS1. And in each case, you excite electrons, and those get passed through a series of electron donors and acceptors, eventually to NADPH. What we saw in the viruses was that viruses had the core reaction center protein, PSBA. And this is an important protein because it turns out 4 Nobel Prizes have been won studying this process because it's how photosynthetic organisms split water to create oxygen and hydrogen. And as you know, we all care a lot about hydrogen and oxygen. Now, the rules of the game were that if you turn the lights out of cyanobacteria, it didn't make viruses. If you poisoned the photosystem, it didn't make viruses. And so these viruses needed photosynthesis in order for those viruses to be productive. Another rule is the host can't make up and repair its abilities because the virus has shut down that capability when it infects. And a third rule is that that PSBA protein, when it creates and splits water, it actually creates oxygen radicals, which damage that protein. And that protein damage means that in any photosynthetic cell, that protein is gone in 30 minutes. And in the oceans, it takes 12 hours to infect. And so these viruses, we think, have this gene to encode that protein, which falls apart in the photosystem. So they have this gene purely to make more viruses. It's selective for the virus. But it also means at any given time, 30% of the cells in the ocean are being controlled by a virus protein. There are many other genes, and I could tell you those stories someday. Viruses steal selectively other pieces of the photosystem, and it's really interesting what their stories are. It's typically whatever is a rate-limiting step to the key metabolism, which drives photosynthesis in this case. So what did Tara do? We actually knew all that before Tara. Tara discovered that viruses also steal central carbon metabolism genes, sulfur cycling genes, nitrogen cycling genes. In fact, there's so many AMGs in viruses that we now know their impact is far more than just photosynthesis. And we're interested in exploring that in a very comprehensive way. So this is actually a map where the gray is all of known metabolism in prokaryotes. And the blue is what we discovered here are the metabolic pathways that microbes use. And a third of those pathways viruses are targeting directly by stealing genes and directly augmenting that metabolism. So that's a big deal. And so we've been working now with these really exciting new community metabolic models to just ask, is this important? And so there's a lot of pictures I would have loved to have shown, but I'm just showing this one. The gray and the blue lines are essentially random levels of importance. And the red line shows the virus signal. And what you can see is there's a giant peak to the right of importance at about 250 level of importance. And so viruses are targeting not only cool metabolisms, but they happen to be the important pathways. When we do further work, we see that these particular pathways drive ecosystem stability. And so viruses are dialing in and manipulating community metabolisms in the oceans in ways that we can actually start to learn about and take advantage of if we have to do the grand geoengineering experiment and start to manipulate the ocean microbiome to help in the fight against climate change. So we can learn a lot about viruses through these sequencing approaches. We've talked about collecting global catalogs, cataloging those and naming those taxonomically through machine learning, the kinds of genes that they steal to manipulate metabolisms, and this approach of studying carbon flux in the oceans. This is all what we call population-based science, and all the tools that we've developed, we've put on an NSF-funded platform called iVirus that allows anyone in the world to do these kinds of analyses. So it's a great model of open science. But I only told you about DNA viruses, and to be honest, mostly DNA viruses that infect bacteria. I'm just going to take 2 minutes to tease you with the RNA virus and giant virus stories. The RNA viruses typically are studied in the context of human disease. And so we had a lot of data to screen. We developed an approach which is 30-fold better than the existing approach using some sophisticated AI-based approaches. The story that we got, among many, was this tree. And this tree shows you in gray all known RNA viruses. In red or pinkish are viruses that were here discovered in the global ocean. And so we actually found 7,000 new RNA virus species. All of them were new species, 99.7%. Many of them fell into all the existing known phyla of RNA viruses, but they also revealed 5 new phyla of RNA viruses. So we doubled the RNA virus sphere that was known from 5 to 10 in one study. This was pretty controversial, and I'm happy to talk with people about the science details, because that's where my heart is. But the key was we did a whole bunch of work on the backdrop of this tree to affirm that phylum level inferences were strong. And when we looked at one of them, which we named the Tara viricota to honor the Tara oceans, it turned out it was widespread throughout the oceans. And it's the key missing link in the early origins of life when RNA jumped into cells. So it's a very exciting new view into life on early Earth. In a similar story, though this is not my work, so I know it much less well, this is another collaborator in the Tara Oceans group that discovered the Myris viridae, which are widespread throughout the oceans. And they ended up, from this phylogenetic tree, you can see, linked to the herpes viridae. And they also offer a link directly to the giant viruses. Interestingly, with respect to early life, giant viruses also have actin proteins, or actin-like proteins, which are thought to be a key hallmark of a higher life. And so these giant viruses sort of blur the boundaries between cellular life and viruses. So just to summarize the virus part, we started counting dots. We shifted instead to using genomic sequence approaches. We have large catalogs now. We've used these in sophisticated ways to establish robust taxonomy, discover the genes that they encode, and their roles in the ecosystems. We've also learned that both RNA viruses and giant viruses from the oceans help us learn a lot about the origins of life on the planet. And this work has been funded by a lot of groups, but particularly 3 different branches of NSF, DOE, and the Gordon and Betty Moore Foundation, which tells you how broad and diverse the kind of work is there. Stepping back from the viruses, Tara really coined this approach of ecosystems biology. The idea of systems biology is to study how a cell works. Tara is trying to do it in the context of cells and all the molecules, organisms, and biomes that are around them. We're doing it studying this diverse around a range of biota, 8 orders of magnitude. And we're doing it in a way that helps develop satisfactory answers to big life science questions around biodiversity, climate change, and the origins of life. And we're trying to really just put biology at the absolute center of physics and chemistry, which is well studied. I'm next going to hand off again to Romain to close.
Thank you. Thank you, Matt. And so you are all fluent in viruses. Like you swallowed these as sponges described by Chris that are close to us. Good evening. So I'm back, yes, and I would like to say and emphasize that we have no idea when we launched this project, this 4-year expedition, the beginning, that we would reach such a novelty. We completely, I think all the group is made of 20 different coordinators, 200 scientists, had no idea that we would go so far into our ignorance. That's really the message I want to say to you, so that we need to go to be curious more and more, to discover better this planet and describe it and try to understand how it will change in the future. So on this vessel, Tara, there is scientists, but also there is a lot of artists on board in residence that they come with us and discuss the expedition that we were doing a long time ago. So now we're doing this again. And also we are talking to a lot of kids, a lot of people around the planet all the time. So this was one of the expeditions we have done. I hope we will be invited by Carnegie again for another topic. Could be on the coral reefs expeditions that we spent 4 years on. The data just came out in Nature in June, last June. Also on plastic pollutions. We also work on the climate change and polar science. I will show you. And also we work, as we speak now, Tara is working on chemical pollutions and biodiversity. What is the impact of chemical pollution that we are running off our lands? So just before I show you the last video, that will be the last 2-3 minutes of voyage of this evening. This was Tara 15 years ago at the North Pole. Bizarre to see a boat in the ice like that, but yeah, that's 1 meter thick, the ice that you see here. It was 3 meters thick 30 years ago. Now it's 1 meter. So we lost 2/3 of the volume of ice. We melted altogether 2/3 of the volume of ice in the Arctic Ocean. And this what you see here, everything you see on this picture is drifting 10 kilometers per day, pushed by the winds. Okay, so you don't do anything, but you travel the world. And this was for 500 days, a year and a half, stuck in the ice, people on board, men and women doing science to understand better the climate change in the Arctic Ocean, as a sentinel of climate change, like a beacon in the dark. And it was so successful that we want to do it again with the technology that you just heard today, what we did on biology. We want to redeploy it in the Arctic Ocean to better understand this very specific environment. What we know so far, based on the data we have, is that this environment in the Arctic Ocean is completely unique. It's another planet. Viruses are different, bacteria are different, everything is different. And we are going to study this for the next 20 years. And the boat you see here is just an image, but now they are welding the aluminum hull over the last 2 months. So we are on it, and it's going to be a crazy journey for the next 20 years. Maybe 10 drift will be going one after one in the coming years. That's for me, and a video that we made for the 20 years anniversary with a lot of numbers and what we have done so far, and take you around the world again. Thank you. Have a good evening.
Quand j'ai découvert le plancton, mais j'ai vu encore la beauté de la terre, la beauté de la création.
On ne soupçonne pas la richesse qu'abrite l'océan, son peuple invisible, si essentiel à la vie sur Terre.
Nous sommes tous connectés. Terre, fleuve, océan.
C'est une révolution, Tara. Cette aventure humaine et scientifique collective.
On est plus fort en étant collectif. Tara, c'est un collectif où chacun contribue.
C'est très beau, Tara. Ça fait comme terre quand on est naufragé.
Depuis 20 ans, nous sommes engagés pour l'océan. Aujourd'hui, nous sommes à un tournant pour l'avenir de la planète, de l'humanité. Il faut aller plus vite, continuer à explorer, à comprendre l'océan, à anticiper les grands bouleversements. Il faut continuer à partager et à changer le monde.
Tout se joue entre maintenant et les 20 prochaines années. Plus que jamais, l'océan a besoin de nous.
I would like to thank you, to thank our partners, of course. Also, I would like to thank Bill Hasseltine, who also was one of the friend who also was important to us to come here tonight. So thank you very much. Thank you for your attentions. And if you have questions, we're here to answer the questions. Chris. Do we have questions?
Yes, let's do our questions now. I'll put in some of my own, and we've got a bunch here on. And keep putting your questions in and keep voting for the questions that are already out there. Let me start at the highest level of this. This week is COP28 going on in Dubai. You're here instead of being there. We should take that as a real honor that you didn't go there. But what, so they're bickering about whether it's, going zero carbon or reducing carbon, which to me seems like it's a little bit of a diversion. What would you say to them about the oceans? What would you say to the folks at COP28 about what they really should be thinking about in terms of climate change? Start there.
Yeah, well, Romain was in the...
Yeah.
I was there.
Oh, you were there, okay.
Yeah.
So even more honored that you rushed over here to come to this. Yeah.
But in the COP system forever, for the last, since 1992, when this thing started, the ocean was not into the game, into the equation. And in COP21 in Paris in 2015, for the first time ever, after 21 years of discussions, people and the negotiators and states acknowledged that the ocean, which is 2/3 of the planet, maybe has a role in the climate change. Maybe 21 years. And so since Paris, now we have the ocean ecosystems as a key player of climate change and a key in both ways. Okay, impacted by climate change, also impacting a lot of climate change. And so now, since then, we are looking a lot of advocacy for ocean protection, saying if we need an ocean alive to sustain the climate, to protect the climate, to absorb carbon dioxide, as you saw. And there is a lot of folks there and on there trying to address this. And given the trajectory of carbon dioxide production, we have a lot to do, of course.
Does anyone else want to comment on that?
Well, I think, that carbon pump is the central point. The oceans are doing something for us, absorbing this gas. And then what happens to that gas is a function of the organisms you heard about tonight. And so I think it's really an exciting time for something like Tara to think about what is the role of those organisms? Are they going to sink or put that carbon dioxide back in?
So there's a lot discussed today about how can we encourage this ocean to store more carbon dioxide? How can we help nature to do more? That's very tricky question. How far we go?
Okay, so I had a question actually for you.
Mm-hmm.
you did these beautiful experiments showing the coexistence or co-occurrence of this complex microbiome, which included both his viruses and the other stuff that's in there. How does co-location necessarily mean interdependent, symbiotic? Do they really need each other? So you talked about distributions, but do they really need each other to survive? Because it doesn't necessarily, just because they're neighbors doesn't mean that they care.
Yes, absolutely.
Yeah.
The co-occurrence patterns that we got is a pure result of a computational exercise, because we have thousands of samples. We can computationally go in and get statistically significant co-occurrence values for the organisms. Now, what are they doing together? Is one parasitizing another? Are they living together in harmony? We don't really know at this stage, because we have so much to explore. So it's basically, a sort of, it's a toy shop. It's a scientific output from a computational exercise to help us understand how organisms may interact with each other.
Yeah.
And our imaging data sets, they help us to ground truth hypotheses, where seeing is believing. When you see one organism inside another, then you know that they're doing something together. So it's the combination of that computational exercise together with the imaging data sets that allow us to discover new interactions between organisms that we had no clue exist. There's a lot more work to be done to really get these interactions into the lab and explore them, study how much are they mutually beneficial, how much are they sometimes beneficial, sometimes harmful, and there's probably going to be a whole spectrum of sorts of interactions that may be more malignant, more benign, more beneficial, that we need to discover.
So are there possibilities that there are like keystone species in there that somehow, without which they're gone? So you want to protect those.
Yes.
The rest of them are sort of hangers on or something like
Indeed.
that. You can think of it that way.
Ecologically, a lot of ecological studies is based on theoretical studies, modeling studies, and we do have fairly good ways to define in an ecological sense what is a keystone species, sort of like the wolf in Yellowstone, right? And we can use those sorts of metrics on our microscopic community and identify potentially those keystone species that particularly important in different ocean environments, and that again provides clues that we can then go dig into and learn more about those potential keystone species to determine their sensitivity to changing temperatures, their sensitivity to acidification, and so on.
Because you see in coral, of course, the bleaching occurs because of change in temperature or salinity, acidity, that the coral, which is symbiotic with one microbe through the photosynthesis, just decides to die, it spits it out. And
that's
so I think that's really fascinating. And coming to your discussion about the interactions between virus and these very important microbes, I was surprised to hear that virus actually have the genome to actually do their own photosynthesis. Is that right? So what actually is going on in that? That's purely symbiotic, I assume, but it could be destructive symbiosis or constructive. I mean, like for human, you get a virus, it can be bad. So can you say a little bit more about how the virus and the microbes bring a healthy relationship to each other?
Yeah, so in the case of that virus photosynthesis gene, there's many proteins in photosystem,
right?
And they have one.
Yeah.
So they probably are randomly sampling the sequence space, and that one gives a fitness advantage for that virus, so it stays in the genome. And then it evolves under different selective pressures in that virus. And so it turns out that sequence actually has a mutation which makes that protein more stable in the photosystem. So you could, if you're a photosystem biochemist, try to take advantage of that virus sequence and say, I'm going to create a better photosystem and use that biology signal. And so, yes, there's different selective pressures, but it's the virus really taking advantage of this larger, complicated framework and just inserting the one weak point.
And is that, I mean, these things do coexist, like all the microbes coexist. Presumably there's a co-evolutionary piece to this, and that's what you're going to discover, presumably, is how they co-evolve to become codependent. Same thing with, if you just look at the microbes. So you just look at the microbes and you look at the microbes plus the virus, is that right? Okay.
We kind of all look at a lot of
Everything,
pieces,
whatever's
actually.
Yeah. So I had another question about distribution, because you had met, there's diurnal cycles from night to day, certainly photosynthetic. Someone once showed me one of the books, you showed diatoms, right? And this beautiful array of gorgeous, as you said, inspired whole architecture and art, right? Someone once told me about, when the person who first drew those pictures, you mentioned his
Burns Tackle.
he did it in a petri dish. And he said he got it all wrong because they were on a flat plane, and these things exist in 3 dimensions. Can you say a little bit about how the 3-dimensional component makes a big difference? So, the sun, if you're up near the surface of the water, you can get plenty of sunlight. If you're down a kilometer, you don't. So can you say a little bit about, the sort of how the 3 dimensions in an ocean, rather than just on the surface and soil, say difference?
Yeah, there's a huge amount of morphological diversity in the ocean, and for a long time this was we didn't really know what that means. It was something that we call the Hutchinson's paradox of the plankton. How can, in a relatively homogeneous environment, how come you have all kinds of different forms of life? Doesn't really make much sense. Now we sort of appreciate that the ocean isn't so homogeneous after all, but there are different sort of spaces in which you can live. There are particles, there are different forms of different materials that you can stick to. Light comes in from the sunlight, of course, but it interacts with the water in different ways depending on the depth. So the different ways that the organisms have learned to intercept the light in optimal ways, in some cases mediated by their morphologies, and there are multiple ways in which the morphology can assist these organisms in the particular lifestyles that they're adapted to. Some of them, they even can exploit effervescent ephemeral gradients in the water, just based on very slight changes in salinity, very slight changes in temperature. Those gradients will actually create a surface on which, if you're the right shape, you can glide on.
Cool.
And these organisms have all kinds of amazing, adaptations to life in this liquid environment.
Cool. So I'm going to stick with the science for a few minutes. Then I want to go to engineering and politics. So let's stick with the science. But from a scientific point of view, you also talked about distribution, and and so if you take the human gut, right? If you took samples from each of our guts, they'd be different microbiomes, 1,000 different microbes, different populations, probably similar microbes with different populations, and yet we all digest the same pizza the same. We die pretty much the same. Is that true of the ocean? If you go from place to place in the ocean, do you see huge variation in the microbiome? And does that variation mean something for the big function, the oxygen production and the carbon sink in the ocean? Is there a big variation when you go from, say, the Arctic to the Caribbean, or from one part of the Caribbean to another part of the Caribbean? What's the variability look like in the ocean?
Do you want to go for that, Matt?
I was going to say, what is the individual in the ocean? We're each different individuals, but yes, the coastal ocean versus the open ocean may share very, very few taxa out of the thousands that each have. But there are gradients, and it does blend together, and it isn't a big water. One interesting thing is that there's only 40% of the microbes in humans, all that have been sampled, as compared to the oceans. And so there's a lot of diversity in the oceans, quite extensive.
Yes, there's the Arctic really is another world. When we look to the microbes, the plankton, we have organisms in the Arctic that have ways of life and that have cellular machineries that are very different to what we find in more temperate regions. It's not just sort of a gradient of life, of adaptations going from the equator up to the polar regions. You really have different kinds of adaptations to the cold. And so as those regions are warming up through climate change, we have a real risk of losing these kinds of organisms
for which there are no other analogues on the planet. So we need to understand how they work better, how they work. Of course, we need to do that in order to hopefully try to protect them better in the future. These organisms living in these extreme environments can also help us perhaps learn more about life on other planets, life on ice planets may well consist of life enclosed in the ice. And so as we learn about these organisms in our polar regions, that can help us learn more about life.
Potential life, like on Enceladus, one of them, you know, I mean, these guys, very interesting.
Yes.
I also want to come back to origins of life in a minute too, so we'll get back to that. So I guess the last question about the microbiome is the soil versus the ocean. So the soil is a very interesting microbiome, which also has lots of variation in population across this country alone, you know, but you can still plant corn in one place and plant corn in another place. How do the oceans and the soil compare in the big picture, but also on the microbiome scale?
There's a lot more diversity in soils than the oceans. In fact, it's such a big problem that if you sample a core in soils, you know, that scale and microscale of change is very, very high. We're a little bit luckier I always say the oceans are pretty easy compared to soils.
Things are drifting.
Even though there's all the, well, but there's still, you take 10 liters of water and you've got a good representation of the diversity there.
I see.
If you take a core of soils, it's actually because we sequence small fragments and try to put them together, the puzzle pieces don't go together well because it's so diverse and we don't.
So metagenomics, right? So,
yes, metagenomics.
Can you just, so the last science question, just to, I mean, Tara Oceans on the ship has been doing amazing stuff, collecting samples. Can you just, so how much of the analysis do you do on board? How much do you bring back to your colleagues in Ohio or wherever? Do you have the omics tools on board that can do this? Do you need to? Because bringing salt water back, you know, there's a risk of losing your species, right?
You know, going at sea, going in the high seas, it costs a lot of money and it's sort of so hard because going in all these countries, you need the permits that takes 8 months to a year to have from the countries. It's a lot of cooperation. It's not easy. So when you are in the ocean, to maximize the sampling, you are completely nuts. How can we take more and more information from this ocean? So 100%, 120% of the work on board is to grasp information. No time for analysis. Barely no time for analysis. So on board Tara, there is no technology, genomics technology. We had some trials with nanopores, but I mean, this is not really serious about DNA today. So everything is taken back to the lab. Every 3 months, there is a shipment from the boat at minus 80 degrees to the labs. And this is where we do the high throughput sequencing, high throughput imagings, and that's what we do all the time. And when we were on the coral reef, we dove 4,000 times to sample the reefs, again, to look at the viruses and bacteria inside the body of the reef, to understand its.
So how do you maintain the same, I mean, you believe that if you have a jar, I don't know what you put it in, a jar of a sample from some part of the ocean, you bring it back to the lab in Ohio, it's going to be the same. It's not, you're not going to find a variation if you keep it at the right temperature and you store it right for however many months.
We basically fix the samples.
And we don't take water back, huh? We filter.
Yeah, we filter the water, get the organisms onto membranes, and then fix those membranes with chemical fixatives. So the organisms are dead, preserved in chemical fixatives, which then allow us to then do microscopy back in the labs and DNA sequencing afterwards.
The honest truth is it was 3 years of arguments about those questions. And we had to do a lot of benchmarking experiments, take fresh seawater, preserve it different ways, figure out what works. I mean, Tara was a pioneer on so many levels, not just preservation, but also like, how do you work in a footprint the size of this carpet to sample all those biology organisms, right?
I mean, if you could, would you have the analysis tools on the boat, or it's because it's out of necessity?
Maybe in the future we could be able to do it with fresh samples, which is maybe more accurate and more efficient. Maybe I'm pretty sure. But what's amazed me that we spent 4 years around the world sampling something we don't see the same way, always the same thing, same routine, very boring, in fact. But the energy on board this vessel for the 4 years that we did this expedition was amazing. Because these scientists that were on board, They were like in the rainforest for the first time with a catching life nobody knows about, and this idea was completely mind blowing. You know, energy on board this boat was completely crazy.
Oh.
Doing the same thing you don't see with your eyes.
Yeah. Very cool.
Bizarre, very bizarre, huh?
So I can't resist one more science question to you about origins of life. You talked about origins of life, you know, at the bottom of the ocean or in the ocean, maybe by vents, some kind of hot vents. But you also mentioned, and this is a question here, the Terra Veracruz. Can you say a little bit more about it for the audience? Because it was a bit technical the way you said it. Can you talk a little bit about why that relates to the hunt for the origins of life?
There was a hypothesis about how RNA got a protein encapsulated form to then jump to cells. And the particular features of the Terra Veracruz gave us 3 signals. And one of them was in that tree that they look like they're towards the base of the tree. And then the other 2 were features about the sequences themselves that suggested these actually are the missing type of particle that we've been hypothesizing and had similar entities of, but had never really seen. So I always say the origins of life stuff is tough because it's really hard to get the full smoking gun, but this is a pretty big smoking gun.
Yeah. No, I think it's a really interesting one that we actually think about too.
Yes, sure.
So let's change a little bit. How much time do we have? Let's change a little bit here to the engineering side. So, you know, there's a lot of talk about geoengineering on Earth. One is to put more clouds up and make the Earth more reflective, higher albedo, so reflects the sunlight. What would you do in bioengineering in the oceans? Just fantasize. I mean, you know, the question is whether you do it or not is you know, there's risks and everything like this. You don't know where it's going to go because it's so complex. But if you thought about bioengineering in the oceans, what would you do?
Can I take this?
You can take it.
Absolutely. If you're this excited,
give our pitch.
Well, I'm an engineer.
Okay.
And a microbiologist. And I'm really excited about this community metabolic modeling. The reason is that it tells you what the nutrients and energy are that need to go through the system. And so if you understand that well enough, then you can try to tune a knob one way or another to get the right output. Just an example from soils is microbes, when soils thaw, can produce carbon dioxide or methane. And methane is a lot worse than carbon dioxide. So could you tune the carbon pathways towards carbon dioxide release?
Right.
And you could do that if you had knowledge about, I need to add this substrate to make these microbes grow to produce carbon dioxide instead of methane.
Right. But the only thing I'd say there is, of course, you know, we all know that we take antibiotics, which can destroy your stomach as well as cure you from a virus or from some bacterial infection you have. So how do you, you know, a lot of people are, how do you know that, you know, we just talked about symbiosis and the interdependencies of these things. How do you know by doing that, there's not a whole chain of deleterious or positive effects that occur? And if you do it in the ocean, you're doing it in the one, it's not a control experiment. You're doing it, you know, it's not like doing it in the lab. So what would you, I mean, how do you think about that, right? I mean,
I think
make these tweaks.
I think that's what Tara is beautiful for, right? Ecosystems biology is let's study the whole thing. And we've learned a lot from the unintended consequences in the sea. There's going to be unintended consequences in these experiments. And so I think if you had the smaller scale pilot to be able to figure out what did I do wrong, that will be helpful. And of course, you're right. There's going to be 5 other methane forms that are going to be a problem. And so we want to learn about what we can before you do the big.
I mean, these things have co-evolved for billions of years, right? So they've figured something out. You guys are trying to describe that. So maybe when you have a better description, you'll be able to say, if I pull this thing out, you know, then that happens. Yeah, I see your face. So let me, we don't have a lot of time left, about 5 minutes, but I wait, which way is that going?
I think it's gone off for.
So we're 5 minutes over already? Okay, we'll go no further than 5 more minutes. I want to ask the question of the political aspects to this because, you know, bioengineering is an example of, you know, there's a risk in it. So people are very risk averse. So what are we going to do? How do any of the 3 of you think about communicating your results to have a positive impact on climate, a positive impact, not just on climate, right? You guys said you're part of the food web. I mean, if without the biome in the oceans and the viruses in the ocean, we'd have no food to eat, right? I mean, it's very plain and simple because of the metabolic cycles. So what would you say? What do you say? I mean, what did you say at COP28, for example, to tell people you have to worry about the oceans? And if you don't, you know, because You even said at the beginning, you can't see what's in the oceans. You can see what's in the forest. You said it. See what's in the forest. So how do you convince people what's in there is important?
What could I say? I mean, we need to. I mean, I'm struck by the beauty of what we saw. That's for sure tonight. So many fantastic, and the magic of this is working very well. This magic to me is, I mean, it's amazing how this is working and producing so much wealth and so much good for the rest of the human beings and the rest of the animals on the planet. And so I think we need to share this adventure, saying we are not human beings, we are not rational, we are emotional animals. And I think we need to tell stories that I told you tonight, because it's an adventure of people at sea, spending their life, giving their monsters at sea, away from their family and from their life. And they give a lot. And they give a lot because they believe in something that is bigger than them. I think this picture, this idea of bringing emotions, showing this magic of life, showing this beauty of organisms, is also a way to better protect them in the future. So I think it's important to use emotions. That's why we have artists on board this vessel, because they are dealing with this emotional fibers.
Bringing scientific knowledge, first and foremost for us as scientists, we try to improve our knowledge of how the ocean works. And then we need to translate that knowledge into actions, into policy actions, to somehow translate that science in a way that politicians can understand. And that's where the challenge is. We need a good science base for future policy issues, of course, but we need, to devise ways to make that science understandable to politicians, and artists are a great means to do that. As one example, but translating the science into policy is a great challenge that the foundation has been working on for quite some years.
Yeah, and we could probably spend 3 hours talking about that problem, especially here, but also in the EU, et cetera. I mean, Tara Oceans does spend time. I mean, this is part of your mission, which is the educational component. So how do you invest from the foundation point of view into sort of this, sort of convening and conveying ideas that you learn?
Well, we invest a lot into the kids, school kids, in the French speaking language. Education is not a question of translation. Education is a question of culture. So we cannot replicate with people what we do in French in the US. It's nothing compared.
Yes.
And so what we do today, we are small in Tara, so we do work on the French language over the world, and we give them food for thought. And we show them, we make them think about what they see and what they reflect on that. And as they are in the school, but in the same time, the star is at sea and doing stuff and trying to understand better. So they're very excited by what they do, because they know in the meantime, they're on the planet, the people are day and night doing this. So they're very excited about it. And this generation today is very afraid sometimes, especially in Europe, because we told them everything.
They're afraid because
they're afraid because we told them everything.
But they also
when I was at school, nobody told me anything about climate change.
They also want action. I mean, these same early career people want action, right?
Yeah, but they're afraid because they are not in the action.
Yeah.
When you are in the action, then you have a
hopefully they don't get jaded and they grow up to be the politicians, and then they.
So they will be in the action in a few years' time when they get graduates.
Okay. To finish, I'd just like to go around to all three of you. I want to start with you, Matt, and just a last word. You may want to say something about a message you want to deliver here, but also if you just want to say, just 2 words about why you're even doing this. What got you into this business of trying to save the world, I guess I should say, but in particular, you're deep into the biology of it. So what got you into doing this?
I just love mysteries.
Okay.
And I love waking up every day and solving a mystery.
And you do that every day?
Every day.
Yeah. Okay. And you play with viruses, so it's a mystery in itself.
Viruses are not all coronavirus. Huh? They are useful.
Yeah.
Chris, what did you?
Just the wanting to learn more about how this invisible life in the ocean works, how it works together, how the functional parts fit together to make a system that works. I find fascinating.
And myself, my commitment I was a dad a few 15 years ago, and suddenly, a few weeks after she was born, I started to think